This is a production of Cornell University.
Good. So what I'm going to do today is talk almost exclusively about undergraduate research. They have contributed heavily on advancing the chemistry and pharmacology and natural products in my lab. So I'm going to present a montage of the most relevant projects that they have undertaken. So I'm going to start a little bit with yeast lipidomics and referring in particular to lipids such as phospholipids, sphingolipids, neutrolipids, and inositides. And then I'm going to spend the rest of the time talking about disease-targeted botanical pharmacology with emphasis on particular diseases, since that is what really drives plant research in my lab. So let me start with a little bit about cell chemical biology. And that's by way of a very early project that an undergrad undertook in my lab. His name was, or is, Matthew Ginsberg. He's an MD degree now. And so at the time that we initiated this project and was financed by a Hatch grant, the egg industry in the state of New York was under heavy financial stress. And that is because regulation was changing, and now small and medium large egg producers needed to accommodate chicken housing requirements, such as larger spaces, et cetera, et cetera. That was putting a lot of stress on them. And so there was a call for grant proposals on research as to how expand the market of eggs and possibly the development of added value products. So this caught my interest because, thinking as a chemist, I look at the egg as a chemical factory, actually. So if one looks at the structure of an egg, it's heavily compartmentalized. The 1/3 of the weight of an egg is the egg yolk. And the egg yolk is actually chock full of lipids, okay? And probably you know a little bit about the stories around cholesterol in eggs, et cetera, which actually have been debunked already. There is no threat of cholesterol intoxication from eggs. So the egg yolk actually is greatly composed of what we call overall as lecithin, which is a mixture of phospholipids. And you're all familiar with the structure of phospholipids, glycerol backbone, phosphorylated with a head group that can be choline, ethanolamine, serine, or inositol, and acetylated by various types of fatty acids. So egg lecithin actually has an application in medicine, and that is the pharmaceutical industry utilizes the lecithin to formulate drugs that are going to be actually administered parenterally. That is, they're going to be administered intravenously, for example. All right. So they're drugs that actually cannot be made water soluble, and therefore they need to be formulated in these vesicles. So egg lecithin has the capacity to form micelles that can then include different types of therapeutic agents. The only problem is that, well, these micelles have had some kind of compatibility with mammalian cell membrane composition. So the idea then was to look at the possibility of deriving from eggs from the state of New York that were appropriate in composition for this application. So I felt the need to put some, economic data into the whole venture as a justification. And just recently, the USDA has established that the value of a grade A large egg is 13 cents. Now, if you look at Cygnus catalog, they sell 250 milligrams of reagent-grade egg lecithin for $59, right? So one grade A large egg contains 2 grams of lecithin worth $472, right? Of course, I'm not including, production costs, obviously. But this is just to exaggerate and make the point, all right? Now, in theory, if you calculate, you can increase the value of an egg by more than 3,000 times in egg lecithin value, right? So, in any event, but the question is, as I said, is this lecithin actually suitable for use by the pharmaceutical industry? Well, I recruited the participation of a New York State egg producer, Creek's Farm Fresh Eggs. Kurt Krier is actually the main owner of the company. It's a family-owned business, actually, but it's a medium-sized egg producer. I think they have like 250,000 layers. So, and he is actually a Cornell alum. So he gladly participated, providing eggs. In addition to providing eggs, he provided also proprietary diet. We felt very strongly that the lecithin derived from these eggs was going to depend on the chemical composition of the diet. So he provided 2 types of chicken feeds that they use in their farms, non-EB and EB, and those are designations that only they know what they mean, right? No, it's proprietary, the diet. So, and then the corresponding eggs produced with the different diets. So the methodology utilized is actually extremely simple. One cracks the egg, separates the yolk, and then the yolk is extracted with a mixture of chloroform ethanol in 2:1 ratio, evaporated to dryness, and prepared for sampling and introduction into a mass spectrometer. So at the time in the core facility, we had a Squire LC electrospray ionization mass spectrometer that one could actually operate. So that was the playground for us for the next 3 years, actually. So it's a very robust instrument, but very limited in capacity of data processing because it's single octopole and ion trap. But in any event, it was very suitable for the analysis of the lecithin. So a lot of Matthew cranked a lot of data processing, and this is basically the results that we receive. Mind you, this is the average mammalian cell membrane lipid composition. The main component is phosphatidylcholine, PC, followed by phosphatidylethanolamine, and then a little bit of phosphatidylinositol. So we characterize the composition profile of the feeds and compare with a sample of commercial lecithin that is utilized by the pharmaceutical industry. As you can see, the non-EB feed has little PC, large amounts of PE, and little PI. However, the EB feeds have larger amounts of PC, comparable amounts of PE, and both probably the feed that compares best with the green bars, which are the composition of the commercial pharmaceutical grade lecithin that contains large amounts of PC, is the one. Now, this is the profile of the lecithin that comes from the chickens that were fed one and the other diet. And as you can see, they vary very differently from the composition of the diets. So the composition that is closest to the commercial lecithin is actually the eggs that were laid from chickens that were fed the non-EB chicken feed, and it's almost equal to the commercial one. Same amounts in PE and very little PI. So if we look at the lecithin composition of the eggs that come from chickens that were fed the non-EB diet is very, very close to the mammalian cell membrane composition. So those were the results that we received, and of course we passed on recommendations. We felt also that the diets can be optimized to better adjust than the profile needed for suitable use by the pharmaceutical industry, given the fact that the diet is very influential in this composition. That's why organic and omega-3 eggs can be produced. You just feed those components to the chicken, and voila, you get eggs that have high content of omega-3s. And just to illustrate that, in addition to the quantification of the phospholipid classes, we actually did a very detailed species profile for each one of the classes. And this is one example. So for each one of the phospholipids, we actually identified each one of the particular species. These numbers represent the added number of carbons coming from the two acyl groups in the actual phospholipid, including its level of saturation. All right, so this actually led to a very robust research collaboration with a group of Susan Henry that to this day it is maintained. And in this particular case, we actually looked at lipidomics and the lipidome of yeast cells, and through the manipulation of different growth conditions and the use of gene delete mutants, we have a better understanding now how these metabolic pathways were actually regulated. So we were fortunate that a newer machine and more capable was acquired by the core facility. We now have a triple quadruple machine that can do multiple reaction monitoring that allows us, in combination with HPLC, to elucidate in one analysis more than 100 lipid species in samples. So this is the lipidome of yeast, depending on the growth stage. We normally work with yeast populations that are in this particular early developmental stage. And here's the whole profile of lipids: cardolipin, sterols, sphingolipids, the phospholipids, neutrolipids, and the lyso products. These, just as an example, the most important phospholipid in yeast is phosphatidylinositol. This is the composition per species of the steady state. However, using now a very innovative use of the stable isotope, we can actually look at de novo biosynthesis of all these species, and therefore, by comparison with the composition of the steady state, infer the level of or degree of remodeling that these phospholipids actually undergo. So this is what we do in combination with all the genetics behind the yeast. So I'm going to move now on to really what I want to spend the rest of the time discussing. That is disease-targeted botanical pharmacology. These are the particular diseases that we're interested in my lab. It looks very ambitious, but actually on a preliminary stage, they're very, very accessible, as I will illustrate. I will concentrate primarily only on cancer and the epigenetics of cancer, Alzheimer's disease, and a more recent line of research that we have, which is menopause. So a definition of botanical pharmacology can be the accumulated and developing knowledge of the effect and mode of action of natural products occurring in plants in the prevention and treatment of disease. And I feel that by studying and investigating the mode of action of a natural product, we are better prepared to provide the pharmaceutical industry with lead compounds that can actually result in a clinical therapy. So I would like to, every time that I talk about a line of research, I would like to introduce the students that participating in them. And as you can see, progress in these type of projects is punctuated and cumulative, and it transcends, generations of undergrads as they move on. And that knowledge and skill and methodological capacity is actually maintained, and new students are trained under it. So some of the early students that I had was Douglas, Ryan, and more recently Chelsea. These two graduate students are actually senior graduate students in programs in pharmacology, Rockefeller, and Cornell, and they're almost done with their PhD work. So to introduce our interest in cancer from the epigenetic standpoint, I have summarized here, the views of the mechanisms of cancer. Now, the classical view is one in which the genome has experienced some kind of change introduced by mutations, deletions of genes, or gene amplifications. And this is the classical view, and the one that has been more investigated and for which there is more information. However, there is an evolving alternative view, and that is that epigenetic mechanisms can actually, and their dysfunctionality, can also result in cancer. So mechanisms by which gene expression can take place is by histone modification, DNA methylation, and non-coding RNA. So these two, histone modification and DNA methylation, actually have been investigated in the context of cancer, and found out that actually they contribute to the unregulated proliferation of cancer cells. And for those two, there are already 2 drugs that are being used clinically to the treatment of certain hematologic cancers. So my interest in particular, and in a moment you will see why, was actually histone modification. Misregulation of histone modification can lead actually to the development of cancer. So just to remind you, histone modification can occur in particular by a mechanism of acetylation and deacetylation. Those changes actually are accomplished by enzymes that we call histone deacetylases or histone acetyltransferases. So acetylated chromatin leads to gene silencing. Acetylation of chromatin leads to relaxation and accessibility to gene expression. So misregulation of this mechanism actually can result in cancer. And here is illustrated. So one particular histone deacetylase that has been identified in a couple of cancers is SIRT1. This is a mammalian histone deacetylase. So it can deacetylase histones, but it can also deacetylase proteins. And illustrated here are 2 proteins that actually the deacetylation of those 2 proteins can result actually in cancer. One protein is p53 that if SIRT1 deacetylates, deactivates, and therefore it precludes the mechanism by which a cell that has a DNA that is damaged cannot undergo apoptosis or DNA repair or growth cell development, and that results in tumor suppression. So if that SIRT1 deacetylase is actually misregulated, it can inactivate or deactivate p53. Or on the other hand, it can actually activate an oncogene like BCL6 that is actually a transcriptional repressor that can result in tumorigenesis. BCL6 and the acetylation of it has been implicated in a particular type of lymphoma. So there is a SIRT1 homologue actually in yeast, and that is SIRT2. So SIRT2 is also a histone deacetylase and actually can silence silence at the telomeric genes at the telomeric region. It's a family of histone deacetylases. SIR2 is the most important, but it forms complexes with SIR3, SIR4. It's a whole that called the SIR-twins, and the acetylation actually silences chromatin. So to study histone deacetylation in the context of finding natural products that can actually prevent their action and end the treatment of cancer, we had a challenge. So before I go on to describe the methodology that we developed to do that, let me just say that these two histone deacetylases small molecules, at a time that we were looking into the possibility of undertaking this type of project, had actually been identified. So and these two chemicals actually were critical in deciding what type of natural products we were going to target, in the possibility that we could set up some kind of methodology to measure histone deacetylase. This particular molecule had been isolated from a fungal isolate that was in the repository of the National Cancer Institute. And it's called splitomycin, and it was found out that it was a histone deacetylase of SIR2 actually in yeast. The other compound is called SAHA for short. It's a synthetic compound, and in 2006 received approval from the FDA to treat this particular type of cell lymphoma. So with this type of chemistry in the background and knowing a little of natural products, one could think of natural product candidates that could be put through an assay to look for histone deacetylase activity. So the question is, where and what to look for histone deacetylase inhibition activity. So knowing that, so using these structures as templates, what came to mind was actually coumarins. So coumarins have this particular basic skeleton. This is a chromone. This is a chrominone. So you can see right away the similarity between the two structures. So we decided that coumarins, because they bear quite close resemblance to splitomycin, could actually be good candidates for histone deacetylase inhibition. So as we know, coumarins are widely distributed in the Rutaceae, Asteraceae, and in Somapiaceae. Now the other group of natural products that were closely resembling SAHA, or actually alkylamides. Okay, so these type of compounds are widely distributed in the Rutaceae, Piperaceae, and some Asteraceae. And as you can see, there is an overlap between the presence of coumarins and alkylamides in the Rutaceae. So Rutaceae became the first target family, to investigate for this type of activity. So the question is, how are we going to do this? So, so here I saw the advice of the Susan Henry group, and decided that, well, if we're going to use yeast to investigate histone deacetylase activity, then we need to actually engineer a yeast strain that will have a reporter gene. And the reporter gene that was decided to use was a URA3 reporter gene introduced in the telomeric region, where the SIR-twins, histone deacetylases in yeast, actually operate. So if the chromatin is deacetylated, there is no URA3 expression. If it is acetylated, then there will be URA3 expression. So we need to translate this theoretical concept into practice. So, okay, so I'm not going to discuss the conditions that are required for this expression and how the reporting is going to take place, and I rather describe how we accomplish actually this. So the yeast strain that was actually engineered was done with a plasmid that was actually provided by Thomas Peets from University of North Carolina. that contain actually the whole right arm of chromosome 5, in which the URA3 gene, reported gene, had been introduced in the telomeric region. So by homologous recombination, we actually engineer a yeast strain that contain now the reported gene in the appropriate place. So now, what is it that URA3 does? Well, URA3 actually is a decarboxylase in the uracil biosynthetic pathway from ortate to uracil that removes the carboxyl group from orodenine 5-phosphate and yielding uridine monophosphate. So now, in this type of methodologies, one can actually give, where the gene is operational, we can give the cells this particular compound, 5-fluoroorotic acid. So this is orotic acid that has a fluorine in this position. And if the cell processes it, it results in the formation of this suicide inhibitor, which is 5-fluorouracil. So depending on whether we allow for URA3 to be expressed or not, we kill or not cells. All right. So now we have a way to actually set an assay to do that. So illustrated here is if the sirtuins, the acetylate, the telomeric region, so the cells can actually grow in the presence of 5-FOA because URA3 is actually non-expressed. But if the sirtuins, the acetylate, and URA3 is actually expressed, then the cells are not going to grow because they're going to form this compound which is actually toxic to them. So that can be done very easily in liquid media, but I have always had a tendency to simplify things for expediency, and what I wanted to develop was a assay on the same basis, but that actually could be used in solid media. So the concept was, if we deposit a histone deacetylase in a filter disc and we introduce it in a Petri dish and inoculate with this particular construct, will it show cell growth inhibition? So here's the proof of concept. Nicotinic acid is a histone deacetylase inhibitor, and you can see that in the presence of it, there is no cell growth, and without it, cells grow. So now we have a system, rather simple, but at the same time complex because of the type of reporting, that we can actually now interrogate natural product chemistry for histone deacetylase inhibition. So as I said, the Rutaceae became the first target family to investigate for natural product chemistry that could actually inhibit histone deacetylation. So at the time, it was almost winter, and so we had to recur to what we had in the greenhouse, and we had actually 2 species of citrus. So we had this particular Rutaceae, Citrofortunella mitis, kumquat, and we had actually limon. So we prepare actually extracts of the leaves of these 2 Rutaceae, and then we put them through the assay. And the result was that, well, the crude chemistry of an methanol extract of this Rutaceae, actually the one from the limon, doesn't inhibit the growth of the cell. So that means that there is no histone deacetylase inhibition, while the extract of kumquat does so. So we pursue then the chemistry of mitis. In this particular case, we were actually targeting, and the reason why we tested methanol extracts is because we were targeting the coumarins in citrus. So we pursue mitis, and so this right away became a big challenge because we could not reproduce this biological activity at will. And every time that we prepared an extract of kumquat and tested, we would either get a positive result or we would not get a result at all. And this was very puzzling. And after reviewing data accumulated, we noticed that the only extracts that were giving a positive result were aged, quote unquote, extracts. And that aging required at least 1 month of sitting on the bench. If we tried freshly prepared extracts, there was no activity at all. So this was actually later confirmed when we started now to suspect that there was a difference between the two types of extracts. Okay, so we applied the chemistry to that. We compare composition, HPLC composition profiles of old and aged extracts, and we found that actually there was a change in profile in which one compound in the that was prominent in the fresh extract, in the 1 month old extract actually decreased in concentration, while another compound actually, or component actually experienced the opposite. Okay, so it started with very low concentrations, and after 1 month it was actually high. This already suggested to us that this change in composition actually was key to the biological activity. We further tested that hypothesis using mass spectrometry, and we found, in effect, there is a compound that in the fresh extract is very low in concentration. It has a molecular weight of 377, and after 1 month actually is high, right? So we went after that compound. We did, bioassay guided fractionation, and that actually resulted in the elucidation of the active principle, that it is only active after the parent compound has actually produced it in large amounts. We don't know the mechanism by which this change occurs, whether it is chemical or biochemical. Nevertheless, it was confirmed. Here is the comparison of the mass spectrum of the parent compound, and this is the mass spectrum of the product that is the active one, which is the result of the loss of these two sugar moieties. So this compound, which is a coumarin, a furanocoumarin for that matter, is actually a histone deacetylase inhibitor. So other students came along, and we targeted then a different rutaceous plant, Santoxylum americanum in this particular case, northern prickly ash, occurs in the area. We collected it. It had been reported that Santoxylum americanum contained 5-MOP, a coumarin, and this particular alkamide, 5-alpha hydroxy sansuol. So we wanted to find out if either one of these two type of compounds actually were histone deacetylases. So extracts actually were prepared, tested. Here is the response, strong response to one partially purified fraction that came from colon chromatography. And we further pursued this biological activity, and it resulted then in a fraction that was rich in a family of compounds which are actually derivatives of cuspidiol, which is a phenylpropanoid type of compound. So we further fractionating using silica gel colon chromatography, gradients in separation. We found fractions that were active, and that by thin layer chromatography showed that it was only one compound. They all contain it. We combined and tested, and the activity is right there. So the compound that was identified by mass spectrometry is actually this cuspidiol analog, cuspidic acid. That's what we're calling it, and it's actually a histone deacetylase inhibitor. So we took a different avenue to fractionation of Santoxylum americanum extracts, and we isolated also an additional histone deacetylase, gamma sansuol, which it hadn't been reported actually in Santoxylum americanum. And so here's the structure. It is actually an alkamide. So, the choosing of the rutaceae, because they combine this type of chemistry that has been shown by analogy to be histone deacetylase, it paid out actually. So we have now, in summary, a few compounds, one furanocoumarin and one phenylpropanoid, and actually one alkylamide. So all this in the course of about 4 years. All right, so let me move on now to a different disease, and this is Alzheimer's. So Alzheimer's is becoming a big problem, not only in North America, but globally, as we have an increasing aging population that it will experience certain incidents of Alzheimer's disease. That in combination with the fact that current therapies in the treatment of Alzheimer's disease are actually not very efficacious clinically. So this interest in Alzheimer's actually started out in 2009 with these two students, Scott and Michael. Both of them already were awarded their MD degrees, and newer students like Cornell and Raleigh. Any event, so this is the neuropathology of Alzheimer's. Very early on, upon postmortem studies, it was actually found out that an Alzheimer's patient brain actually has experienced considerable shrinkage. And neuronal loss mainly occurs in the cerebral cortex and the hippocampal region, where the cholinergic network resides, and the cholinergic network actually is responsible for memory and cognition. So as you can see, affecting these parts of the brain results in loss of those two functions. So histological preparations had also shown that associated with this neuronal loss or the formation of plaques, that now we call them amyloid, beta amyloid plaques, and also accumulation of protein, tau protein fibrils inside of the cells, all right? All that causes Alzheimer's disease. So based on these results and biochemical studies of brains of Alzheimer's individuals, it was noted actually that levels of acetylcholine, the neurotransmitter in these areas of the brain, was actually very low. So the first hypothesis or model proposed for the disease was one in which the Alzheimer's disease was actually the result of a low concentration of acetylcholine in this synaptic cleft between two neurons, right? So acetylcholine is actually made from acetate and choline by a choline acetyltransferase and degraded by an enzyme that is called acetylcholinesterase, all right? So when acetylcholine is released into the synaptic cleft, finds its way into receptors which are in the postsynaptic neuron. One of them, the muscarinic acetylcholine receptor, is actually a G protein coupled receptor. And so it was then proposed that that signaling cascade that resulted from that interaction actually was affecting the processing of APP, which is the protein that actually is the precursor to the beta amyloid peptide, which aggregates into the plaques. So the cholinergic hypothesis that was proposed in 1975 blamed low amounts of acetylcholine in the synaptic cleft for the production of the beta amyloid peptide. So from this model, it was very clear what the solution was. Well, then develop a drug that could actually restore normal levels of acetylcholine in the synaptic cleft. And that target was actually the cholinesterase, the acetylcholinesterase, the enzyme that degrades acetylcholine. So the first and only generation of actually drugs, what they do is they block the activity of acetylcholinesterase, restoring normal levels of acetylcholine in the synaptic cleft. And clinically, it was predicted that if that was the case, then downstream, the production of beta amyloid peptide was going to decrease and the progression of the disorder was going to slow down, right? So these are the drugs that are in current use. So the cholinergic hypothesis was proposed in 1975. The first drugs that came into clinical use were Aricept and rivastigmine. This compound is completely synthetic. This is actually derived from a natural product, physostigmine, that occurs in a African legume, which is actually very toxic. So the other compound actually, or the other acetylcholinesterase inhibitor approved in 2004 is galantamine, that actually occurs in a host of plants. I just cited daffodil because it's very timely right now to talk about that. And it is used as it is found actually in these plants. There is an alternative treatment that blocks actually the receptor of glutamate, which it is thought to contribute to the formation of the tau protein fibrils in the neurons. And this is all that exists in clinical use. And their efficacy is actually extremely poor, right? So, given the interest that we had in this, and I'm going to start to abbreviate because I'm running out of time now. So we decided to look for acetylcholinesterase inhibitors. So the question was, what type of plants are we going to look for to screen for acetylcholinesterase inhibition? So the plants that we decided to target were plants that have been claimed to possess neurotropic activity. And neurotropics are defined, as agents that can be or act as memory enhancers, neuroenhancers, cognitive enhancers, etc. And the ethnobotanies are rich in those types of descriptions. So the question now was, how are we going to measure this? Well, we had actually a fluorometric assay to do it, but it just was it was not very practical to use it. It's very good at quantitating activity and gathering kinetics, but not very quick for targeting a particular component in a plant. So we opted to use an alternative, which is a bioautographic assay that can be developed in a thin layer chromatogram. It entails the resolving the components of a mixture on TLC, applying the enzyme, and then a reagent that generates a background and contrasts with a discoloration, which is actually an indicator of acetylcholinesterase inhibition. And here's the proof of concept. This is the assay, and this whitish discoloration here is actually Aricept, the drug that is used clinically. So now we had an assay, and we decided then on what plants we were going to target. So one of the first plants that we actually targeted was Maca, Lepidium ayeni, an Andean brassicaceae that traditionally is used for the treatment of male infertility, but that it lists as another benefit actually improvement in memory and brain function. So this is the assay as it looks. So we diagnosed that these areas of discoloration in particular were due to alkaloids in Maca, as we got a positive with Dragendorf reagent, which is highly specific for alkaloids. And we went on to isolate those components. And I'm not going to discuss in depth, but these are the types of alkaloids that are found in Maca. They're very unique, imidazole type of alkaloids. Surprisingly, Maca, this brassicaceous plant, contains also alkylamides, and they're all acetylcholinesterase inhibitors. So it could be the foundation for the claim, that Maca probably benefits memory and function. So another plant that another nootropic plant that we targeted also is lemon balm, Bellis officinalis in the Lamiaceae, that there is a claim that alleviates mild Alzheimer's. So here is the acetylcholinesterase inhibition assay. These are the spots that we targeted. There are others, but we felt that this was the major component. It's a phenolic compound, as it gives positive with ferric chloride. And ultimately, we decided that very likely this compound was rosmarinic acid. We run HPLC analysis, compared with an authentic sample of rosmarinic acid, and sure enough, it's there in the extracts of the plant, and confirmed by mass spectrometry. So, so these are in summary, these are the acetylcholinesterase inhibitors that we have isolated: one alkylamide and two alkaloids from Lepidium ayeni, and one phenylpropanoid in Melissa officinalis, rosmarinic acid. Okay, so lastly, I'm just going to briefly discuss menopause and our interest in menopause. And actually it's an extension of the Alzheimer's disease interest that we have. So participants in this project or research is Marianne, Kamal, Jeanette, and Heather. So we had interest in actually phytoestrogens for a number of reasons. One of them was very pragmatic, and that is, well, this particular group of plants have been utilized for a purported estrogenic activity. These two, black cohosh and red clover, have been utilized in alternative treatment of menopause symptomatology. These other two actually have been used traditionally as galactogos to increase breast milk production in lactating women. So we felt that these two types of effects were caused by the presence of estrogen mimics. So, but at the same time, we felt that there was no really good hold on the estrogenic chemistry. Chemistry had been described, but never conclusively demonstrated that those reported compounds were the phytoestrogens. So in a revisionist type of approach, we decided, okay, let's look again at these plants. We have now an interest. We have plants to look at. All we needed now was to find out how can we do this. So, so menopause is a age-related biological phenomenon, results in the reproductive and the loss of the reproductive capacity of a woman, and is caused by gradual decrease in estrogen production in the ovaries. So, as we reviewed the literature, and we're getting ready to investigate estrogen mimic activity, we realized that estrogen was actually integral to a number of symptoms associated with menopause. So they're the classical ones, and they're the result from that decrease in estrogen and decrease in progesterone. But one thing actually caught our interest, cited amongst the umbrella designation of psychological symptoms, was actually poor memory. And that caught our interest. So how is this? I mean, what is the relationship between loss of memory or decline in memory and estrogen? Well, there are a number of studies that actually have conclusively demonstrated that there is an association between or correlation between the progression of menopause and the decline in memory and cognitive function. So this is one of the more recent ones, published in 2013, conducted at the University of Rochester, which a group of middle-aged women in different stages of menopause were actually tested physiologically and psychologically for verbal learning capacity, verbal memory, fine motor skills, and attention working memory. And as you can see, these bars represent the women, the groups of women in the different stages. And as you can see, especially in verbal memory, there is a decline associated with progression of menopause. And it's true for all of the other functions that actually were measured. So here we have actually a very concrete evidence that there too are associated. So now mechanistically, why is that? Well, looking at the literature, we now been able to summarize some of the brain functions that estrogen actually performs in the brain. So estrogen actually is a regulator of the enzyme that biosynthesizes acetylcholine, right? So the enzyme is choline acetyltransferase. So, so this enzyme is under positive regulation of estrogen. So the other function of estrogen is envisioned as regulating the processing of APP and via the modulation of the enzymes that cleave APP. And in the presence of estrogen, the amyloidogenic pathway that leads to the formation of the amyloid plaques is actually downregulated. And the third property is actually, if you notice, A ring of estrogen is aromatic with a hydroxyl group. This compound is antioxidant. So it performs also a neuroprotective function in the brain. Now, if you remove estrogen from the brain, all this happens. Okay? The protection as an antioxidant is lost. The upregulation of chat is lost. Less acetylcholine is produced. And it actually increases the risk in a woman to develop Alzheimer's disease. And we know very clearly the statistics. Women are at twice the risk of developing Alzheimer's disease than men. Right? So this actually led us to formulate a different approach to the study of estrogens, and because of the role of estrogen in menopause. And what we're looking for now is what I call combinatorial pharmacology in plants. That is, chemistry in plants that can act as an antioxidant, that can act as a promoter of acetylcholine formation, and that can act as an estrogen mimic to reduce the risk of developing Alzheimer's disease, right? So we have encountered now actually those plants. This is how we assay for estrogenic activity. We engineered another yeast cell with this particular plasmid that contains the human estrogen receptor and a beta-galactosidase reported gene. So the plasmid is introduced in the cell, and we have now an engineered yeast cell that, upon the stimulus of estrogen or an estrogen mimic, actually triggers the expression of beta-galactosidase that process OMPG to OMP that we can measure then spectrophotometrically. Here is the proof of concept. Such construct treated with estrogen results in a positive response, all right? So the first plant that we actually approached for study is fenugreek, a Fabaceae, used since antiquity for a number of reasons, but is used actually as a galactagogue prominently to increase lactation in women. So I'm just going to go to the results and the chemistry that we have actually isolated and identified conclusively. So one estrogenic component of fenugreek is actually dalsgenin. We isolated, characterized it, and evaluated its estrogenic activity. It's in the chloroform ethanol extract. And in the aqueous ethanol extract of fenugreek, actually, we isolated 3 isoflavones, which are actually the same 3 isoflavones plus present in soy. And we know that these isoflavones, have a estrogenic mimicking capacity. So they're present also in fenugreek, and the presence of this chemistry is actually responsible for the estrogenic activity. We now have defined a larger group of plants to study. The junior undergrad students that are working in these projects have already established estrogenic activity, antioxidant activity, and acetylcholinesterase inhibition activity, and they're in the process of isolating the active principles in this plant. All of them in the Lamiaceae, primarily salvias, which are or have been shown to be estrogenic. Now I'm going to stop there. I just want to acknowledge. See, I knew that I had too many slides. So this is a summary of chemistry that is estrogenic and acetylcholinesterase inhibitory. So the isoflavones and diosgenin, the estrogen mimics in fenugreek. And another plant that we have studied is adjuvant, Trichospermum ami, in the Apiaceae, that contains estrogen mimics, phenolic acids, all of them, which also show acetylcholinesterase activity. I would like just to acknowledge the rest of the undergrad students that have worked in my lab throughout the years. I didn't get the chance to talk about their projects in chemical ecology, malaria, diabetes, and cancer. All of them already in graduate school. Thank you for your attention.
This has been a production of Cornell University, on the web at cornell.edu.